Welcome you all in the second session of day two for our 1 week online FDP on innovation in pharmacy education and application of AI, enhancing teaching learning practices in collaboration with Society of Pharmaceutical Education and Research. For this, I would like to welcome our speaker, Dr. Gaurav Goyal, sir. Firstly, good afternoon, sir.
Good afternoon.
Before proceeding further, I would like to introduce our today's speaker. Dr. Goyal, Dr. Goyal is currently working in the position of Deputy General Manager and heading Transdermal Formulation and Development Department at Ghasidas Life Sciences, Ahmedabad. He has total 15 years of industrial and academic research experience and worked with well-known industries such as Silpa Medicare, Ekums Drug and Pharmaceuticals and Rusan Pharma Limited. Sir also has academic experience and guided number of students at ICT Mumbai, ISF College of Pharmacy Moga and MET Institute of Pharmacy Mumbai. Sir had completed his doctorate and master's studies in pharmacy from Institute of Chemical Technology Mumbai and graduation in pharmacy from Jamia Hamdard, New Delhi. Sir has developed the first indigenous generic fentanyl transdermal patch, which was commercialized in the year of 2009, and later many more transdermal technologies was developed by the Sir. Sir has focused research area on the development of oral thin films, transdermal therapeutic systems, implants, nasal sprays, lyophilized injectables, sustained release topical and oral solid formulation. Sir is the lifetime member of Indian Pharmaceutical Association, that is APTI, and Control Release Society Indian chapter. Welcome you, sir, at ISFCP platform.
Thank you very much. I am within my family members,
Yes,
so
sir,
I am very
definitely.
happy and delighted to present my presentation on this FDP to all the faculty members. Uh, it is my esteemed privilege that I am still, you people are inviting me and recognizing me at this particular platform. And I wish this is useful for the all the faculty members who are on the online at present and are listening to me. So today I am going to present on transdermal drug delivery system because this is the field where many companies now, earlier it is only one or two companies who were there in India who uh, who are actively involved in production of transdermal systems. But in late 2 or 3 years, many companies like Urbindo Pharma and many more companies are there. And Cubicicles in Mumbai, they have started their manufacturing plant in India. So, so there is a very big scope uh, for the researchers as well as the students. They should understand this particular technology. And in fact, this is the platform where all the most of the faculties are there. So I think it will be useful for them. And those faculties who are really very interested to learn the transdermal technology, they can invite me to their colleges and they can collaborate with me. And then now there are two or three companies, Indian companies, who have started manufacturing of transdermal equipments. So that is very good part, and their equipments are also not very costly, somewhere around 5 lakhs, 6 lakhs rupees only. So they, on your any of the projects, you can, uh, I think, buy those equipments, and that can you can start with some transdermal technology where a student can get a exposure, a lab exposure, I can say. But definitely, it will be very helpful for them to get placed into the industries uh, easily. So, with this note, I just start with my presentation. You can ask the questions later on, or in between, also, you can ask the questions if any. So I just share my screen.
जी सर, जी सर.
Yeah, just a minute. Is it visible?
Yes sir, yes sir, yes sir.
Okay. So today's topic for discussion is advances in transdermal drug delivery systems. So there are in this particular presentation we are going to discuss on the generations of transdermal systems. What is the role of chemistry in the development? How these transdermal systems get manufactured and tested into the laboratories, and one of the practical example how to calculate the skin flux and certain regulatory guidances on the transdermal patches. So as we know, this transdermal delivery system delivers a drug from the uppermost layer of the skin, that is epidermis, to the blood vessels, whereas the topical drug delivery systems only supplies up to the dermis. The drug does not deliver into the blood circulation. So transdermal systems, basically the maximum extended drug delivery systems patches which are approved by FDA is 7 days uh, for the transdermal drug delivery system. So there are potential advantages of transdermal drug delivery system, in which we can see this when we take the tablets, it is a spiking is there above the maximum safe concentration MSC, and certain times it comes below the MEC, that is minimum effective concentration. Whereas the transdermal system maintain the drug delivery in a zero order controlled release manner. It is reduces the toxic effect because the blood the levels do not generally do not go above the toxic level. It avoids the fast metabolism of the drug, which is very helpful for the adherence of the treatment, because once you apply the patch, it remains on the skin for the 7 days for the prolonged period of time. So you do not need to get adhere. They need to take the multiple dosing of the formulation, and interruption is very much possible. If you want termination of the therapy, you can easily can remove the patch yourself, the patient can remove the patch, and then the therapy can be terminated. It avoids the dose dumping. For all these faculties, maybe this is the new terminology, I just say some few lines on this. Dose dumping is the phenomenon like when we take the sustained drug delivery system tablet. So when it goes into the stomach, and if anybody consuming the ethanol or alcohol, so maybe the sustained release matrix get disturbed, or the integrity of the matrix destroyed, and the dose, the complete drug which is there for the 24 hours or 12 hours, is released immediately. So this type of problem is known as dose dumping, which can even lead to death of the patient. But as is the transdermal systems are applied on the skin, so if anybody is consuming alcohol, it will not leads to dose dumping. So that is the importance of this particular drug delivery system. So in fact, if anybody like if from the start, I can say the transdermal system is just like a bandage. Like before applying the bandage, we remove a release liner from the bandage, and then we apply the drug matrix on the skin. But bandages are basically for the topical. They do not deliver the drug to the blood. But in the transdermal system, it is similar, but it delivers the drug into the blood. So it is almost like a bandage, where you can see a uppermost layer which is known as backing layer, then the drug adhesive layer, and the release liner. So this is one of the patch of Ghasidas, which is norelgestromin and ethylene estradiol, which is shown onto the screen. Just to make you to understand, just it is like a bandage. It is this particular patch is rectangular shape. It can be have circular shape. Any design you can make it. So basically, the first generation, we there are four generations. And now almost the fifth generation of drug delivery system for the transdermal is under development. So fourth generation transdermal systems is already well manufactured, well established, well developed, and their systems are available into the market. But the first drug delivery system, the first generation, it is the diffusion based. That is the passive diffusion based. It is not the active diffusion. It is generally because of the concentration gradient from the patch to the skin. It leads to the transportation of the drug molecule, API molecules across the skin layers, different skin layers. It is basically a diffusion based, a passive diffusion based drug delivery system, and therefore if it is passive. So the molecular weight of the drug should be low. The drug should be having the good lipophilicity, and it is efficacious at the low doses. These are the three essential properties of the drug candidates for the partial passive diffusion. There is a small video.
TDDS, Transdermal Drug Delivery System, also known as patches, placed on the skin to deliver a specific dose of medication through the skin and into the bloodstream. TDDS involves layers of the skin. The outermost layer is the stratum corneum or epidermis. The layer below this is the dermis. There are also the blood vessels, which are on the subcutaneous layer. How does the drug get from the skin into the blood? Once the TDDS is applied to the skin, the drug is then released and travels to the stratum corneum, then to the dermis, then migrates to the capillary vessels and into the infected area. Now let's go to the lab. Let's formulate a transdermal drug delivery system. The bottom layer is the liner. It protects the patch during storage and is removed prior to use. Next is the drug solution, which is in direct contact with the release liner. The following layer is the membrane. It is used to control the release of the drug. Next is the adhesive. It serves to adhere the components of the patch together along with adhering the patch to the skin. Lastly, the backing. It surrounds the drug and protects the patch from the outer environment. So why pick TDDS as your way of medication? It is easy to use, more improved and convenient patient compliance. Self-medication is possible. Avoid gastrointestinal tract absorption problems for drugs. Avoids first-pass metabolism. Rapid termination in case of toxicity is possible. Avoids in-drug fluctuation drug levels. Minimizes undesirable side effects. Reduces frequency of dosing. Maintains therapeutic level for 1 to 7 days. Controlled delivery results in reliability and predictable blood levels. Substitute for oral route. But how come TDDS is not always preferred by patients? Local irritation. Uncomfortable to wear. Allergic reaction. Drug requiring high blood levels are unsuitable. Significant lag time. Limited skin permeability. Difficulty and long time adherence. Restricted to potent drug. Drug with long half-life cannot be formulated. Daily dose of more than 10 milligrams is not possible. Cannot develop for drugs of large molecular size. Did you know that the first adhesive transdermal delivery system was approved by the FDA in 1979? These are the scopolamine patches for motion sickness. This method of delivery became widely recognized when nicotine patches were introduced in 1991 for smoking cessation. Nicotine patches deliver a constant dose of nicotine across the skin that helps to relieve the symptoms which are associated with tobacco withdrawal. 4 million deaths every year are experienced by the USA due to cigarette smoking. This makes nicotine patches popular and wide selling in that country. In 2014, the DOH in the Philippines distributed contraceptive patches to promote adolescent health and family planning. According to PNS market research, transdermal drug delivery systems market is forecasted to cross $60 billion in size by 2023. The use of transdermal patches is the highest in North America owing to heavy expenditure by the government on healthcare and supportive reimbursement policies. as well as in Europe and in Asia Pacific. The market for transdermal drug delivery systems is expected to observe the fastest growth on demand in China and India due to the improvement in healthcare infrastructures and rise in the healthcare expenditures in developing countries. This growth is characterized by growing aging population, the number of patients with neurovascular disorders, increasing prevalence of chronic diseases, growing concern about painless drug delivery, rising adoption of new drug delivery system technologies, and benefits offered by transdermal drug delivery systems. USA, Australia, Canada, Europe are most stringent when it comes to global regulatory requirements for transdermal drug delivery systems. This includes standard skin irritation, sensitization, adhesion studies with the patch itself in animals or humans. Thank you for watching. We hope you enjoyed and learned a lot from this video.
Uh, so this is a very illustrative and easy to understand presentation made by one of the research scientists. Uh, Coming again to the formulation considerations of the transdermal system, one of the important consideration is API, where the regulatory requirement is it should be the minimum residual drug should be there. What does it mean that like this after application? Because it is a passive diffusion. If we are incorporating in a transdermal system like 4 milligram of the drug, but within like in 7 days, generally it is 30 to 40% of the drug gets uh, permeated, and rest of the 70 to 60% of the drug remains into the inside the patch. So when it is a so this 60% of the drug can be abused if it is belongs to the narcotic category or psychotropic category. Like fentanyl is one of the example where there was a lot of abuse of this particular drug because fentanyl comes under the category of narcotic. So this is the uh, all the countries have the principle that the minimum amount this residual drug content should be there after application. Like after if it is a 3 days patch or 7 days patch, after application the amount which is left inside the patch it should be minimum. And pressure sensitive adhesive that it should be having the optimum tack level, optimum peel level, and it adhere to the skin for the wear period of time, and it should provide the chemical and physical stability to the but API. Antioxidants generally we need to incorporate if the drug is highly sensitive for the oxidation. Solvents, generally organic solvents are used for the manufacturing of transdermal patches. Certain times we need to use a rate controlling membrane between the two layers. We generally have the two layers, multi-layer transdermal patches, and between the two layers we incorporate the rate controlling membrane so that the rate can be properly controlled. Zero order rate can be properly controlled. Release liner is the component which is there into the transdermal system for the easy removal. Like before application, we need to uh, remove the release liner from the patch and then apply the patch on the skin. That plastic part, which we generally removed, it is known as the release liner. The important quality consideration that it should be easily removed from the adhesive polymer. That is the important consideration. The backing membrane is the topmost layer of the patch. Generally, it is tan color, which in bandages, if you can see, it is a tan color cloth-like structure. So it should be flexible and should be breathable so that oxygen or gases can permeate. Otherwise, after if it is a 7 days application, your skin can die, your skin cells can die. So therefore, oxygen it should be permeable. The backing membrane should be permeable enough so that there should be an exchange of gases, which is possible. And then pouching laminate is the pouch in which we generally pack the patches, and they generally provide the chemical and physical stability. So these are the different components which are used to manufacture the transdermal system. Coming to the physicochemical properties of the drug candidate, so I have made it to make you to understand very easily and with certain examples of the drug molecules which are These scopolamine patches, clonidine patches, estradiol patches, fentanyl patches, nicotine patches, rivastigmine and buprenorphine patches, I have taken the example. So the general criteria is a dose should be less than 20 milligram. So where all, if you can see, all these molecules complies to this particular criteria. Half-life should be less than 20 hours. So one of the molecules, all these molecules comply to this criteria except buprenorphine, where the half-life is higher, but still the transdermal patch of this molecule exists because of its advantages. Molecular weight is less than 400 daltons, and buprenorphine is one of the exception. Melting point should be less than 200. These are the preferable criteria, general criteria for transdermal molecules for the drug candidates, where all the molecules comply except buprenorphine. Partition coefficient between 1 to 2, and if you say only estradiol, which is 4.2, which is almost on the upper limit border, all molecules comply to this particular criteria, general criteria for drug candidates. Aqueous solubility should be more than 1 milligram. Estradiol does not comply, otherwise all other molecules comply to this criteria. pH of the aqueous saturated solution should be between 5 to 9, where all the drug molecules comply to this. This is the general criteria for the selection of the molecules. So if any molecule, if you come across a very new molecule and you want to try that whether we can use the whether we can make the transdermal patch of this particular molecule or not. So this is the general criteria which needs to be taken in consideration. If it is matching all matching to the general criteria, falls within the general criteria, we can use it for the transdermal. We can explore for the transdermal application. Correct. So polymorphism should be evaluated and justified in terms of product performance. Polymorphism to be evaluated because generally the API exists in different polymorphs. So which polymorph is more suitable for the product performance permission, that we need to check out. If illustrative, this is the simple illustration of manufacturing of the transdermal patch, where we generally make an adhesive mask in which we incorporate the adhesives. The adhesives are of transdermal patches are known as pressure sensitive adhesives, which is known as PSA, pressure sensitive, because just when we apply the pressure, it sticks to the skin. So generally, in this category of pressure-sensitive adhesives, the general molecules, general material of construction are silicones, or either acrylates, or either polyisobutylens. These are the three classes of molecules which can be used for as a pressure-sensitive adhesives. Other two classes which are not very much explored commercially, it is the one is the polystyrene, which is rubber based. This is one of the class which is not very much explored for the manufacturing of transdermal patches, right? So we use the adhesive mask and incorporate the drug into this, and then we coat on a release liner, and then sandwich between the release liner backing membrane and make the wide rolls of this, and then we cut those wide rows into the narrow rows, and then convert these narrow rows into the patch. So this is the die cutting to individual TDS systems we generally receive. In laboratory, this is the laboratory machine which people can use. So this is a small video here which I can make you to understand. This is you try to understand this. On the release liner, we coat it and then we laminate it. So this is the release liner, and these are the different parameters. Where it is the surface temperature and the you can see the blower speed. Above this machine, you can see one pipe. It is not above the machine, which is for the evaporation of the solvent, because it contains the organic solvent, ethyl acetate and naphthalene. You can see the pipe above. So the scientist is applying a blade for a uniform coating. So he is making the surface flat first of all, and then he is going to apply a blade which is known as the doctor blade, and maintain a distance, the gap between the blade and the liner, so that the wet coating thickness can be controlled properly. So he's adjusting the thickness through calipers, and these are drying where the RPM is 1000 RPM. So now he's going to pour the adhesive mass on the liner. The blade is coming and the coating of wet coating done, and then this wet part, coated part, goes inside the machine for drying purpose. This particular assembly will go inside the machine, and then after drying, it is a set parameters, the critical process parameters, the drying temperature and the drying blower speed, where it dries and the solvents get evaporated and the film becomes dried. Then after the drying, we laminate with using the backing membrane. So here it is only release liner. On the release liner, we coated it, and then this will go inside the uh, container, right? So this is lamination after drying. It is getting laminated using the backing membrane. So this particular presentation contains various videos to make you to understand how this transdermal system works. So above there is a backing membrane. So this is the laminator. This is the below is the coated laminate which is there. From the above the backing membrane it is sandwiched between the two layers. So you can see the below layer is moving from below and the upper layer is coming from top, and from behind the laminated patch comes. So after this lamination, we convert the patch into the individual rolls. Like this is the laboratory setup. In this laboratory setup, we uses the dies to cut the patch into the respective shape and size. So this is a solid die. You just see the patch is cut and now it is converted. Right. So one of the very good example. This is one of the examples from my PhD research work, where I want to show how transdermal chemistry plays a very important role in the manufacturing of transdermal systems. So generally, these polymers adhesives, which are known as PSA, pressure sensitive adhesives, they have the Hansen solubility parameter from the literature. So silicones have the Hansen solubility parameter of around 11 to 13 megapascal, 18 to 20 for acrylates, and fentanyl for 20.6. So if you see fentanyl is somewhere near to acrylates, that means fentanyl has good solubility into the acrylates, and there is a gap difference of 7 between the silicones and fentanyl. So the solubility of fentanyl is less in silicones. So what happens if we make the fentanyl transdermal system into silicones? The problem is that maybe it will crystallize because the solubility of fentanyl is not poor into silicones, but it is as fentanyl has the very close Hansen solubility parameter with acrylates. The solubility of fentanyl is very high into acrylates. So because of that, it will the problem of crystallization into the transdermal system with acrylates is least possible. So that is a good system, but in transdermal manufacturing, we have to understand that the drug molecule, the API, that is here in this case it is fentanyl, it should not having the very good solubility with the polymers. Otherwise, polymers will not allow the molecule to permeate inside the skin. It will retain the molecule within its matrix. So therefore, if you see below the examples, if we make a with a polyacrylate adhesive, we need to load the drug very high. It is almost 4.2 milligram drug we need to incorporate into the patch to get the in vivo release of 25 microgram per hour for 72 hours. It is a 3-day patch. 72 hours means 3 days. So the drug release rate is 25 microgram per hour. For this to get this particular drug delivery system from the patch system, we generally need to incorporate 4.2 milligram of the drug. But if we use silicons, which this is my research project, Rusan, where we use silicons, uh, where the problem of solubility is less. Therefore, I use the mixture of silicons, and we have reduced the quantity per patch from 4.2 milligram to 3.0 milligram to get the same drug release profile, in vivo drug release profile. So in this first one of the slides previously, I have told you the residual drug amount should be low. So in case of Johnson and Johnson Duragesic patch, the residual drug amount after application 3 days it is 2.4 milligram. Where in the case of Rusan, which we have developed, it is 1.2 milligram, which is almost half. So this is the important point to highlight. If we can use different polymers to achieve the residual drug amount lower. So here, as I told you, I have used the mixture of silicone pressure sensitive adhesives. Why I require to use the mixture, that is I want to highlight here, because silicones are of two types, standard and amine compatible. In my system, this fentanyl is it contains the amino group, NH2 group, as a functional group on the drug molecule. So this NH2 group in the standard polymers, if you see, it is contains the hydroxyl group at the end chain, OH groups, which you can see here, right? And OH group react with the NH2 group, and they do with the hydrogen bonding. So they have the hydrogen bonding between the OH and NH2 group of uh, hydrogen of the amino group and hydrogen of hydroxyl group, so which leads to the good solubility into the standard bio PSA. That is very good. But what happens uh, if we but what is a different problem with this? It is having the very good solubility into the standard bio PSA, but it reacts uh, with the drug molecule, and the drug release is very retarded. So what we have, we used amine compatible, where we find this the hydroxyl group is not there, but the drug release is very fast. And in fact, drug release is very fast because it is not there is no hydrogen bonding between the fentanyl and bio PSA. And moreover, as I told you, there is a problem of crystallization because the solubility of fentanyl is low inside the silicons, as far as the Hansen solubility parameter, which we have seen in the previous slide, this slide. To make it soluble, we used a standard polymer because of the hydrogen bonding, the solubility of fentanyl is increased into the bio PSA. So to get the combination, then therefore we have used the combination of standard and amine compatible. Amine compatible we use to get the release profile, and standard bio-PSA we use to get the solubility, sufficient solubility of phentalate to the silicon polymers. So this is one of the reasons we have used the combination of standard and amine compatible polymers, silicon polymer, pressure sensitive. So these are the CQAs, like in tablet we have disintegration, dissolution rate, right? In the similar manner, in which we have the control of transdermal system on the basis of CQAs. The various CQAs which are there into the transdermal system is the appearance, It should be looking good. It should be lack of visible crystals. The crystals should not be present. Dimensions we measure for the transdermal patch. We do the assay. We find out the impurities and the degradation products which are there. Uniformity of dosage unit, patch to patch variation should not be there. In fact, in this, not only the API, up to the shelf life, we need to measure the enhancer, permission enhancer content and antioxidant and preservative content up to the shelf life. It should remain within the specification limits. So for this, we also need to develop the assay method for these three excipients if they are into the formulation. Then there are physical tests like peel adhesion. It will remain is peel adhesion. We generally do it with the in vitro test. In my next slides, I will cover this how we are going to do this. So peel adhesion is the force which is required to peel off the patch from the skin. Release liner peel strength, as I told you, release liner has to be removed from the patch before application. So release liner peel strength should be low enough. Tack is the force which is required to apply the patch. So tack should be high. It is immediately should stick to the skin. But tack should not be so much high. Like if you apply the patch on the skin and it will not remove, because if by chance you have applied at the wrong position, it should come out and then you able to apply on the another skin. So tack should not be very high, but it should not be very low also. Shear is the displacement of the patch from the its location. Like certainly, if you apply on the patch on your biceps, and in the morning if you wake up and then you find out the patch is displaced from the biceps, and that is not a good patch. So therefore, we find out the shear. Cold flow is the property of the polymer. Generally, if you see on the sides of the edges of the patch, there is certain adhesive which oozes out. So the cold that is known as cold flow. So it should not be there. Generally, in research work, we generally keep the patch inside the book or a copy, and then just find out on the another day or after 10 days, the adhesive should not stick onto the paper. It should remain inside the patch. So this is one of the like in universities or in labs, we can do this test by keeping, like in childhood we used to keep leaf inside the papers or books. In the similar manner, you can keep a patch inside the book and just find out whether how much polymer is ooze out. In this, we generally do the in vitro drug release testing, like we do dissolution testing for tablets. So there are USP 5, 6, and 7 apparatus, which I'm going to cover into my next slides. We drug substance. In this, two microorganisms which are very much important is the Staphylococcus aureus, and we need to identify where they should be absent into the transdermal system, because these otherwise they can cause skin infections. And we also control these residual monomers, because it is a polymeric system. So the monomers which are there inside the polymer, it should be very low, otherwise it can cause toxicity. So this is one of the examples where I'm going to tell you how to calculate the flux. So this is a France diffusion cell. I think this is known as the receiver compartment, and one is the donor compartment, and in between we use the skin membrane or artificial skin, and then we apply the patch on this, and then or membrane, or either it will be you need to apply from the, and above this particular skin you apply the patch, so that the from the patch the drug permeates inside the skin or membrane and goes into the receiver compartment, which is present below, and we calculate the how much drug is diffused from the patch inside the receiver compartment. So these are the HPLC readings. Generally, in some Indian papers, previously I found out that they do using UV, which is not the correct method, because by UV you will most of the polymers get leached from the skin, and they show the absorbance interference. So it is not a very specific method. It loses It fails into the specificity. Therefore, we should use HPLC system whenever you do some transdiffusion studies using animal skin. If you are using some artificial skin, then UV can be used. But if we are using some animal skin or human cadaver skin, we should not use the UV apparatus. And then we calculate, these are the absorbance test readings using HPLC. From this test readings, we convert into the concentration and divide by the area of the skin. So but in the denominator we use the area of the skin like centimeter square. So we found out the how much amount is permeated in microgram per centimeter square in different time intervals, and this is a cumulative. This is not the cumulative; it is the concentration. But when we convert into the cumulative, it will something like this. So 0.60, which is the first reading, and then it added into 0.222. Then you will get to the reading, next reading. So you add up the previous time points absorbance or concentrations. So you will get the cumulative permeation. So on the x-axis you plot the time, and the y-axis you plot the cumulative concentration. You will get a slope, a graph, the slope of which is known as, that is y upon x. Y upon x, that slope of this particular line, it is known as the flux. So the average flux of this particular transdermal system is 0.0372 if we calculate by this particular data. The instruments which are used, okay, very interestingly, all the faculties which are present here, I would like to make you to understand this, because in I have seen certain this conceptually, people are not very much clear about this particular thing. A dissolution method or drug release method, like if you have the sustained release tablet of 24 hours, so you can have the dissolution method of 6 hours also. That is acceptable, because dissolution method and drug release method, which are in vitro methods, generally it is for only for QC release, to check the batch to batch variations. It is not something relation with the in vivo. How this performance, if your in vitro drug release is 24 hours, we cannot predict it in vivo system, that in the within the inside of the body it will remain and it will release for 24 hours, because the diesel media you have selected, because you have selected the RPM of the pattern. So it is not going to, the correlation should not be very much necessary. You can have the dissolution media of 6 hours or dissolution method of 6 hours, but it can inside the body it can release the drug up to 24 hours or vice versa. Similarly, like for the 7 days patches, we can have the method of 6 hours and 12 hours drug release method, because drug release method you try to understand, it should not be having if the patch we have releasing 90% of the drug in 6 hours, it doesn't mean that it will remain on the skin also, it will release 90% of the drug within 6 hours. It can release the drug up to 7 days. So it is why we do then the drug release method, only to check the batch to batch variation. It is a QC release testing. Whenever we release the batch, we do some testing. On the basis of those testing, we release that particular batch. So it is only for the to check the quality of the transdermal system, we do the drug release test. Otherwise, transdermal system, whenever we do the development, we need to develop through the France diffusion cell, because in this we used RLD, reference listed drug, and then in comparison to that, whatever test formulation test product, you have to compare the flux of those formulations. But for this, you need not to be having the same drug release profile which the innovator is having, RLD. RLD can have different release profile and we can have different release profile. I hope this is clear to you. So in transdermal system, the basic, very important thing, because in tablets we generally match the dissolution rate. We do the F2 testing, that is the similarity factor testing. But in transdermal system, we compare the flux. Our flux should be similar to the RLD or innovator product. That is very important. So this is the USP5 apparatus, which is paddle over the disc. In the USB 6 apparatus, it is just like a cylinder apparatus where you apply the patch on the cylinder part. Then the adhesive side, the drug side, should be facing towards the dissolution media. Similar in the sink, this is paddle over the disc. In this disc, we apply the patch from the upward side so that the adhesive surface should face towards the paddle. There is USB 7 apparatus, which is the reciprocating type of apparatus, just like a USB 3, in which we apply the patch and it goes up and down. It will not revolve, it goes up and down. So this is the reciprocating type of USB apparatus, from which we can find out the drug release. This is one of the apparatus which is known as universal tensile tester, from which we can find out the physical addition parameters. So I just show you how this transdermal system works out. You can see this is the patch which is applied on the stainless steel panel, and it is peel addition. It is peeling the patch in the 90-degree angle. This is the shear test where it is a patch is applied on the stainless steel panel, and we are finding out whether it will be displaced or not. What is the displacement force which is required? This is also one of the 180-degree peel addition testing. If you see, the patch will be 180-degree removed from the patch. Earlier we saw 90-degree, it is 180-degree experiment. Correct? Yeah. So this is all about the first generation transdermal systems. Now we come to the second generation transdermal systems, where people started using chemical penetration enhancers and using some active forces like antiphorosis and some ultrasound waves, which is known as, that technology is known as sonophorosis. So using these things, the big molecules, the big vaccines or big proteinaceous molecules, which is having the higher molecular weight, can be actively transported across the skin membranes. In the third generation, people use destructive hyperthermia principle, where they have transported the vaccines, peptides such as insulin, parathyroid hormones, influenza vaccines, and many more things using application of the skin on the this particular principle is known as thermal ablation, where they destroy the upper layers of the skin by passing the electromagnetic radiations, and which helps into the transportation of the molecules across the skin. So in this, their particular but it can lead to the removal of stratum corneum, the uppermost layer, without damaging the deeper tissues. So this is certain destruction of the above tissues, that is the uppermost layer, but it will be very helpful if the person is suffering from cancer or they require such type of therapy. In the third generation, it is also microneedles and electroporation people are exploring. Now, microneedles research on microneedles and USP, the United States Pharmacopoeia, also going to include the chapter on microneedles. So this is going to be very, very big. People are doing a lot of research on microneedles, and it will be the future of. It is the future also in the transdermal field. So people, I think the researchers should start working on this. If they today, if they are going to start, whatever help required from my side, just let me know. We will collaborate and we can do this type of studies. The fourth generation, which is the artificial intelligent transdermal drug delivery system, where it is a chip-based system. It will sometimes microneedles will be permitted, like you can handle, you can wear like a watch on your skin, and then some microneedles will go inside your skin and can monitor the from your blood. They can sense, they can diagnose your blood, and then can find out whether your blood pressure is high or low, and then immediately can release the drug. Basically, it is from the transdermal system inside the blood. So it is the absolute type of microchip based system where like the glucose level, if your glucose level goes up, it will deliver the insulin more inside the blood. When your glucose level is normal, it will stop delivering the drug. So it is a delivery based assisted device assisted delivery based system, where if you require the drug, it will deliver. If you don't require the drug, it will stop. If so, it is uh, device-assisted transdermal drug delivery system. So this is the future generation, and in US, a lot of people have started working on this fourth generation transdermal drug delivery systems. Thank you very much for your attention. If you have any questions, just let me know.
Okay, definitely, sir. If any audience want to ask a question, he or she can. So I think, sir, this is the era of AI. So I wish to know that nowadays in India, the companies like Ghasidas or other giants are working in this area in transdermal section or not. They are dependent on conventional or shifted towards the AI.
Yeah, it's a very good question. Like the fourth generation transdermal delivery system, it is the artificial intelligence based systems. That is why one of the very important delivery systems, and Jadis is also involving into the development of AI based systems. Like in the last slide, we covered this. He through the microchip based systems, it can AI can able to identify what is the level of uh, components into inside your blood, and then can deliver the drug accordingly. So it is a device assisted drug delivery system. It can monitor the glucose level, it can monitor your blood pressure, and then can deliver the drug accordingly as per the requirement.
Sir, as I have seen that you have elaborated the minimal invasive system in third generation. So this will also be incorporated in the fourth generation because it needed the minimal invasive system.
Correct. It is a the fourth generation. It is actually micro-based system. The third generation, it is the minimal invasive system. So the fourth generation uses the third generation systems also, because microneedles is definitely required to be like, because the needle should be go inside your skin and should not cause any much more pain. So therefore, fourth generation use the third generation system also along with the chip based systems.
Okay, thank you, sir. So I would like be grateful to summarize this knowledgeable lecture. So, sir has covered advances in transdermal drug delivery system, including different generation from first to fourth generation, and construction of. Sir has displayed a one interesting video that displayed the construction of transdermal drug delivery patches and a specific advantage, disadvantage, and some regulatory aspects. Formulation consideration and drug candidates with favorable criteria for transdermal drug delivery system, and also displayed manufacturing processes. And another interesting video was there that displayed the coating and drying at laboratory scale process. And sir has nicely explained the transdermal chemistry also, why some polymers chemistry including that are important for the transdermal drug delivery system, and some control aspects, how to evaluate what are the parameters to evaluate the transdermal drug delivery system, and some instruments used in the evaluation process, that was USP apparatus 5, 6, and 7, and universal tensile tester, and also describe the second, third, and fourth generation of transdermal drug delivery system. Sir, ISF College of Pharmacy is thankful to you for your valuable time you have spared at our platform. I think our audience will be benefited from this lecture in terms of transdermal drug delivery system. So thank you again, sir, from ISFCME family. Thank you, sir.
Thank you very much. Thank you to the ISF family. Thank you to principal of the college, Dr. GD Gupta sir. Uh, and thank you to you to invite me on this particular
Thank you, sir.
platform, faculty development program. I hope the faculties get benefit from for this. At least they will get the understanding of transformer system. And if anybody wants to connect with me to understand more on this, they can contact me anytime uh, through you. You can share my email address with them. That is not a problem with me.
Yes. Thanks again, sir. Thank you very much. Thank you
Okay.
all the
Thank you.
participants who are joined here and online. Thank you everyone. Have a nice day. Thank you.